next speaker. Okay, so it's a laser pointer. Okay, great. Anna Devor is sort of a, I think, known to many of you, sort of a pioneer in, optical imaging methods to understand kind of the fundamentals of neurovascular coupling. I think it's actually literally true that I think 2 textbooks, at least that I have, literally had to be rewritten based on Anna's work. And I can show you, I've got the 2 editions, which we can compare. I think she's contributed, enormously to our understanding of this sort of neuronal basis of, or the hemodynamic basis of the fMRI signal. She's the editor-in-chief for the journal Neurophotonics. She's also the head of a large brain initiative study that I've the kind of privilege to be a part of that is investigating the kind of underpinnings of human fMRI signals. And so it's a great pleasure to have Anna here today to talk to us about a neuronal specificity of hemodynamics. Thank you so much.
Hello, ladies and gentlemen. How's everybody doing? Good? How's the course so far? Good. Wonderful. So I'm very happy to be here talking to you. Thank you very much for the super kind introduction. Just to mention, there are many other people involved in this research that I will touch upon now. And also welcome to Boston for those of you who are out of town. So we are going to talk about the kind of the biological underpinnings of hemodynamic responses, and most of the studies are done in animals. So now why in animals? Well, because this is where we can get to all these microscopic bits and pieces and be very specific. So there are lots and lots of tools that are available to us in animals that are invasive tools and cannot be translated tool-wise to humans. Sorry, I'm hot. So what we do is that we look at all these different types of neurons, at neuronal circuits, at microscopic vasodilation and vasoconstriction, at microscopic oxygen consumption, and then we try to build models that help us to translate this microscopic bits and pieces into more macroscopic picture, and also kind of bridge from animals to humans. Also, just to mention that there are those macroscopic variables that are measurable in terms of non-invasive imaging. So with functional MRI, we can look at macroscopic CBF and CMRO2. Cerebral metabolic rate of oxygen. You already heard about this, or this comes first time? Okay, well, so this is cerebral metabolic rate of oxygen, CMRO2. And also we can talk about non-invasive electrical measurements such as MEG or EEG, where we can predict feed-forward electrical type. So I kind of structured this in terms of a few questions that we can address here. So the first one is, in general, what is the mechanism underlying positive and negative volt? Okay. And I'll start with introducing to you this optical imaging that's based on hemoglobin absorption, that many times is called imaging of intrinsic signals. And this is because there's no extrinsic probes required. So this is based on the fact that if you look at the absorption of oxy and deoxyhemoglobin, so this is here in red oxyhemoglobin and blue deoxyhemoglobin, they differ, okay? And because they differ across different wavelengths, we can sample few wavelengths and calculate oxy and deoxyhemoglobin. This is in mice, for example, or in rats. You can do this relatively minimally invasively. So you can do thin skull, for example, or you can remove skull and have a window. And this is going to be an imaging of usually the kind of the dorsal part of cerebral cortex in these animals. And what you do is that you get those images per wavelength. And then there is a way to compute from this. We're computing oxy, deoxy, and total hemoglobin. So this is HbO, Hb, and HbT in here. And obviously, if we define some region of interest, we can also talk about time courses of those signals. So normally for a positive hemodynamic response, you see an increase in volume. So this is going to be an increase in HBT, total hemoglobin in here, right? Because if there is more volume of blood, there is more hemoglobin. We also see an increase in oxyhemoglobin and decrease in deoxyhemoglobin. So that's a signature of our normal positive bolt. So this is a study that we did long time ago when I just joined as postdoctoral fellow here in this building. So in here, we just stimulated one single whisker. So this is the world's smallest response to the smallest sensory stimulus. And these are time series of oxydeoxy and total hemoglobin. So one feature that I would like you to pay attention to is that this response is kind of centers around. Warm colors in here is an increase. So this is an increase in oxyhemoglobin, also increase in total hemoglobin. But there's this decrease in the surround. And this is not unlikely negative bold that has been seen in many human experiments as well. So here's an example of negative bold in human S1 that was recorded about the same time. This is for median nerve stimulus in humans, and you can see that there is a positive bold in here, but there's plenty of negative bold kind of surrounding the same hemisphere, but also in the opposite side, ipsilateral to the stimulus. And if we look at this as a time course, there's obviously a decrease, okay? Here's another study. So in this one, we can also see that there is, for a visual stimulus in this case, there is a positive bold surrounded by some negative bold. So one can argue that this presence of negative bold in some way is, just the usual kind of feature of our hemodynamic responses that we observe both in animals and in humans. So here is a seminal study by Nikos Lagathitis that combined electrical recording. So they had an electrode that was recording at the same time when they did bold functional MRI in monkeys, in this case with the visual stimulus. And they showed that when they kind of move the visual stimulus around in space such that this electrode records once from the region of positive bold and then from the region of negative bold, you can see that the spiking activity follows the sign of the bold response, okay? So when there is an increase in spiking activity, there's a positive bold. When there's a decrease in spiking activity, there's a negative bold. And the idea was, well, most likely this is due to neuronal inhibition. Okay, so to look at this a little closer, I need to introduce to you yet another technology, and this is two-photon imaging. So two-photon imaging is a microscopic imaging technology that we can use in animals. For this, we usually have to remove a skull and put a window so that we see more clear. And here's an example. This is a window. It's a cortical window in a rat in this case. And with two-photon microscopy, we have this absolutely exquisite resolution, including in Z, in depth. And this can be thought of as kind of like a voxel of your functional MRI. And if you look at this, at the complexity of this vasculature, it's sort of overwhelming. But also remember that these black places in here are not just void, right? So this is where all the rest of the tissue is. So neurons are there, astrocytes, microglia, all these wonderful things. Also, when we talk about two-photon measurements and application to vascular kind of imaging, we need to mention David Kleinfeld. So he's my dear collaborator, also part of the same program that John mentioned before. David was the first one to, in 1998, I believe, to apply two-photon microscopy that at that time was a very freshly developed technology to imaging of vasculature, in particular blood flow. So what can we do with this? Well, so we can ask a question, what kind of actual vascular kind of dilation and constriction corresponds to positive and negative bold? And the way that we did this at that time is that first we would map on the surface, we would map response to a sensory stimulus. So the one in here would be the center. And we did this with just surface potentials. And after that, we would look at dilation of all those different arterioles at different locations relative to the center. And so you can see that close to the center, you can see this is the baseline. So here the stimulus comes, there's a dilation, there's a little bit of constriction going back to the baseline. But if you are far away from the center, you're going to see much bigger constriction before it goes back to the baseline. So that was the basic finding. So now I can show it to you in a different way. So now these are all these responses organized dependent on where they are. And so now I'm going to normalize the initial dilation here, and we're going to look at the constriction. Ready? So here we go. So now all the dilations are of the same amplitude, and you can see how big the constriction is far away from the center. The center is in here. When we kind of move further away. So this prompted us to kind of make many of those measurements, and then try to sort them by the distance from the center. And when you do this, it becomes very clear that if you sort them by the distance and then normalize the initial dilation, you can see that constriction, relative constriction, gets much larger as you move to the surround. And these are those places where we see negative gold. So the other interesting thing that came out from this study is that we tried to do some fitting to fit all those different responses with a formalism that basically was a weighted sum of two functions. One is positive, the other one is negative. These are, sort of alpha functions. One is positive, one is negative. And if their peaks are not at the same time, if you sum those guys, you get something biphasic. So we tried to fit the parameters to be able to fit this data, and we got a really good fit. So now a question for you. What did we prove by this kind of fitting exercise? Take 2 hemispheres of your own brain, massage, and think about that. So what do you think that we proved in this experiment? Did we prove anything about positive, negative gold?
Depends on the distance from the center.
It depends on the distance from the center, but in terms of the mechanism of this, or in terms of the actual kind of, we didn't prove anything at all. So we just proved that we know how to do MATLAB, so this is true. But in addition to this, nothing at all. However, this is an exercise that suggests something. And so what it suggests to us is that there may be these two different forces that act at the same time. So there may be that there's one force that tries to dilate, and there's another one that tries to constrict, and they're always active together. And depends on what exactly their time courses and amplitudes are, who is winning and who is losing, you're going to get this kind of biphasic thing that sometimes is going to be mostly dominated by dilation and sometimes constriction. So up to now, we know that positive gold is correlated with functional excitation and predominant dilation because we're at the center of the response where excitation is the biggest. And negative is with functional inhibition and predominant constriction. I'm also kind of trying to go through this relatively quickly. So there are lots of more details, including voltage imaging of actual neuronal excitability in the corresponding papers. When you guys go back, if you want, all the citations are in there, and so you can take a closer look. So now the next question was, is dilation and constriction, are they driven by vasoactive metabolites? And so the idea at that time was that it's the accumulation of metabolites that actually cause hemodynamic response, okay? And the question was, well, so is it that or is it something else? So is it the products of metabolism that drive the response? So to address this, what we did at that time is that we decided to look at the response in the contra and ipsilateral hemisphere for the same stimulus. So what we're doing here is that we are recording from certain locations as before, but we present the stimulus once to one side and once to the other side. In this case, it was a 4-pulse stimulus, so one versus the other. And you can see that on the ipsilateral side, there's mostly constriction everywhere. So, okay, so there's constriction on the ipsilateral side, there's dilation on the contralateral side. What about uptake of glucose? So for this, we did 2DG radiography to look at the uptake in glucose. And to make this story short, this is little, complex plots to take a look at. So I'll just point your attention to this hump in here. So this red hump in here shows that there is an uptake of glucose also on the ipsilateral side. So this is all published and there are lots of details in it, but the punchline is that there is a lot of uptake on the contralateral side, but there's also a lot of uptake in the ipsilateral side. And this is not new, at that time it wasn't new because people showed with the direct electrical stimulus. So this is from Bruno Weber in Zurich. And they showed that if you do direct electrical stimulation, you also see that there is some uptake on the ipsilateral side. All right. So in addition to this, there was also this very interesting other study that was published about the same time, a little later. This is from Ute Lindauer, and she's in Germany. Where is she? In Berlin, I think. And what they did is that they repeated this type of experiment in a hyperbaric chamber so that there's lots of oxygen that gets in the blood, more than normal, okay? Such that doesn't matter what kind of demand you have, you still have lots of oxygen. And the idea was if somehow these are the availability of oxygen or metabolites that cause this hemodynamic response, in this case, because there's so much oxygen, you would just block the response. It's not needed because the oxygen is so abundant. And the answer is so it's still there, exactly the same. So it's not the availability of oxygen that drives the response either. All right, so some interesting conclusions, but I just gave you the gist. So this is from her paper in 2010. And so now if the dilation and constriction are not driven by metabolites and not because of oxygen sensing, so the only other thing which is left really in there is signaling. And it can be either through neurons or some other cells. I'll show you the bit of neurons. And we also did some study of astrocytes, and they're not involved, but we can talk about this later. Let's focus on neurons. So for neurons, we and others have been using optogenetics from the time of the inception of this technology. So optogenetics is wonderful. You can introduce the optogenetic actuator or inhibitor in certain types of neurons. In this case, it's all the inhibitory neurons in the mouse cortex. And then you can play those games. So what you do is that you excite them all, and then you ask a question: what happens to the vasculature? Does it dilate? Does it constrict? What happens? And so here how it looks. This is a little 2-photon imaging frame that goes through a vessel. The vessel is not exactly straight, so you see a bit of shadow, but every time when there's a flash, so that's the optogenetic stimulus. And then you can see that there is dilation, then there's a constriction, and then it goes back to the baseline. So if we were to take a look at this as a time course, it would look like that. There is a baseline, there is dilation, constriction, and back to the baseline. We can also do sensory stimulus. So for the sensory stimulus, we are very close to the center of the response, so there's not much constriction. When we just stimulate inhibitory interneurons, right? So there's no glutamate, there's no, maybe astrocytes responding to glutamate, or none of this is present in here. So we're just looking at activation of inhibitory neurons and inhibitory only. We can see both dilation and constriction. And in this study, we also were able to identify what actually were receptors responsible for constriction. And these are the Y1 receptors for NPY. So here we're blocking these receptors, the constriction goes away and dilation is still there. What happens if we just excitatory neurons? Well, this is this red one. So the red one is dilation due to excitation of pyramidal cells. And this is biphasic response due to excitation of interneurons. So cortical pyramidal cells drive dilation. Inhibitory neurons can drive both dilation and constriction. but at least they're the only ones that can drive constriction. So here's one interesting point, that cortical layers that do differ in their neuronal composition. We'll look at this in a second. And the next question is, what about the laminar specificity of the whole thing? So first, I need to mention that there is this kind of specificity to different cell types, neuronal cell types in this case, across different layers. Okay, you can see that there's a fair amount of overlap. So this type of interneurons that are blue kind of present everywhere, but there are lots of them at the bottom, and there aren't very many at the top. One other thing to keep in mind, that this type of classification that you're going to see in other papers is based on the location of the cell body. If you're looking, so for example, this is a pyramidal cell, okay, and this is our layers. So this is layer 1, layer 2, layer 3, 4, 5, and 6. Okay? This is a pyramidal cell, this red one. And although it would be classified as layer 5 because the soma is in layer 5, well, but the apical dendrites go through the leaves all the way up, right? So this is a caveat to keep in mind, an important one. However, one can still think in terms of kind of like densities and the clouds of densities of axons, if it's an axonal release of vasoactive messengers. And you're going to see that there is some specificity across the layers of that too. But there's one other thing to keep in mind. And so the thing to keep in mind is that both dilation and constriction propagate along arterial walls. So here's a paper from Beth Hillman some 10 years ago plus. And so what she showed in there at the surface, that this is a piece of the surface that has both the hind paw of a mouse, I think, or a rat, and there is a forepaw, okay, in the somatosensory cortex. So this region corresponds to forepaw, this is hind paw. So she can stimulate either or, and look at the direction of vasodilation propagating wave in both cases. And you can see that indeed there is a wave, and it propagates in opposite directions. So this is a demonstration in vivo, and this is why it is, this paper was very impactful at that time, and still is. But people have shown long time before that if you just take an artery and you put it in a dish, you can apply some dilators or constrictors on one end, and there's a propagating way all the way through. So vasculature has its own little brain. So there's a mechanism in there that just propagates the whole thing. And if you think about this, it makes sense, because let's say that you have some neuronal activity at one place and it starts somewhere maybe deeper down. You have to open then an entire path. So you have to have this upstream vasodilation towards the peel surface to really decrease resistance nicely so that there's a lot of blood flow through. So on that note, the same thing also holds for penetrating arteries. So there's a propagation all the way through. So there's a study we also did some years ago, and in there we looked at the propagation along the penetrating arterioles. And you can see that there's this kind of wave that goes through. So all arteries, arterioles have this ability to propagate signals up and down. What about the laminar specificity of CMRO2? Well, so that is an easier target, so the laminar specificity of dilation is difficult because of this propagation issue. Laminar specificity of blood flow, by the way, is different in a sense that if there is more capillary density in a certain layer, there's going to be more blood flow through that layer. So this has been shown. In addition to this, CMRO2 is happening in mitochondria, right? So mitochondria are the ones that metabolize oxygen. And so this is super local to the microscopic location of mitochondria. So if it happens to be in a spine and, layer 4, well, so it's right there. So the very last technology that I want to mention to you very quickly is this 2-photon imaging of oxygen. This is something that we can use to estimate CMRO2 very locally across layers. And this is, it's a 2-photon imaging. It's a lifetime microscopy. I'll show to you what it is in a second. And so this is based on this probe. So there's an extrinsic probe that changes lifetime of phosphorescence of this probe because oxygen quenches this phosphorescence. So more oxygen, there's less lifetime. And with this one, we and others did this beautiful, beautiful mapping of intravascular oxygenation. So you can kind of go through the entire capillary, bed and figure out what the oxygenation is. You can also inject it into the tissue and get this sort of like waterfalls of oxygen availability around diving arterioles. I have no interest in Bruker, but they're selling our instrument. So what can we do with this? Well, with that caveat that this probe has to be delivered, but after we have a way to deliver this, we can have it in an awake mouse, and we can go through the layers. We can measure oxygen sort of landscapes in there. And after that, we can feed a model to feed CMR2, to calculate CMR2. So this is a little bit of technical details. Let's not worry about that. And so let me tell you about the model. So the model is really simple. We are getting away with literally a murder. So we are taking, looking at this very complex biologically, asymmetric cases, and we feed it with a model of diffusion from single cylinder. Why does it make sense? Because if you look from the top down at penetrating arterioles, there's this kind of hole of vascular capillary density around those arterioles. Why? Because there are lots of oxygen leaving from artery. And angiogenesis, which is the process of growing of vessels, capillaries in this case, is oxygen sensitive, so it's not needed in here, so there are no capillaries, okay? And because of this, we can treat it as this kind of cylinder where this is going to be our diving arteriole and the oxygen is leaving from it to feed the tissue right around. So now when we do this, there is some formalism. We can talk about this later, but we showed, some time ago that with this type of model, we can look at those kind of gradients of oxygen availability from arteriole to the tissue. And we can calculate CMR2 by feeding this data to this type of model. And so now when we do this across layers, so this is our CMR2, estimated CMR2, and this is cortical depth. And we're doing this in a mouse which is resting, so it's not doing anything at all, and we're looking at the somatosensory cortex. My original idea was, hypothesis, that the biggest CMR2 would be somewhere in layer 4. So in my history of my research, every time when I had a hypothesis, it was the other one. So I never, ever had a case where I said like, okay, I really think it should be this way, and we do experiment and it's that way. Never. So in this particular case, the biggest CMR2 actually was in layer 1. If after that, when we thought about this for a while, and we did lots and lots of control experiments trying to understand, whether we're missing something. So I think that this really depends where we have our mouse. So this is an awake mouse that just sits in a cradle and does absolutely nothing. We're calling it resting state. Under these conditions, there's no stimulus coming to layer 4, right? And layer 4 in the somatosensory cortex really reserved for the thalamus. So this also tells us that one important thing that we need to do is that we need to repeat this experiment across different behavioral or, engagement states. And this is something that we're busy with doing now. So this paper is published. So this is kind of the conclusion from this, is that CMR2 does vary across cortical depth, but also it is very likely to be dependent on the type of computation performed by the brain or on the brain state. And I don't have time to talk about this, but I just want to throw it there so that you maybe, would look at this on your own. So there is also aerobic glycolysis, which always is going on. And it's kind of like a dark matter of energy consumption for us because we don't have good probes to estimate this whatsoever. So some of the energy demand is met by CMR2, but some of this is glucose, which is glycolic, so there's no oxygen. And this one also is layer dependent. And not only that, but it's driven by actual neuromodulatory neurotransmitters such as norepinephrine. So this makes the whole thing super interesting. And we're working very hard trying to get to that dark matter question. So layer resolved functional MRI is something that we definitely need. It's really, really important to develop instrumentation to be able to do this, not only in humans, but also in animals. And at the end, there is some possibility that it can be used to infer the brain state and the well-being of neuromodulatory circuits. And if we could get there, this would be a super cool thing to do. So this is, oh, I see. So this is the end. I just wanted to, there are lots of people that contributed to this research all the way through the last 20 years of me doing this, 25. This is our current lab at Boston University. So I'm very fortunate to have all these wonderful people in the lab. I'm also very fortunate to have, together with John, this wonderful project that we're part of. We just had this retreat and Peter was there, gave a keynote talk. We enjoyed this tremendously, so this is how happy we were. And if you're still not believing it, so this is how happy we are. So this is me, John is right there, and here's Peter. Ladies and gentlemen, thank you very much. And if any questions are there, so I'll happy to